All right, last one on the playlist. Yay. Okay, so proteomics, or comprehensive proteomics to be more specific. So this stuff is very, very simple. The word proteomics in and of itself just means studying proteins. But in our case, this is comprehensive proteomics, as in we're studying proteins by looking at them all at once. So in the previous two videos, we were aiming for specific antigens or protein probes, but in this one, we're studying thousands of proteins together. We're able to look at many proteins at the same time. And this concept is described in one word: unbiased. All right. Another name for this whole technique is comprehensive proteome profiling. I'm not going to bother writing the entire thing down, but it's just comprehensive proteome profiling. And it can actually work in one of two ways. It either finds proteins by trying to recognize and find certain peptide sequences in the sample. We all know what peptides are. They're amino acid sequences. So it looks for a specific sequence, and then it calls, if it finds a specific sequence in a protein, it calls it protein X or protein Y. Or it could just try to recognize the entire damn protein by looking at the full size thing instead. Okay, so now that that is out the way, I will consider the what part done. Now let's look at how it's done. There aren't many details, but the point is, you take a sample and you put it in this huge machine called a robotic station. This huge magical pit that just gets the work done for you. We call it a robotic station, and there's actually a picture right here. You can find it in your third lecture. Anyway, the point is you have three main steps. The first one being separation, shown right here. I'll explain what that is in a minute. And the second one being analysis. There's also another picture that represents analysis, but I thought in our case, let's just focus on having one picture so you don't get confused. And finally, finding out how your results can be valuable on a clinical level. I'll just put a question mark to represent that. Okay, so in separation, the robotic station is going to separate the proteins into teeny tiny spots in that using gel electrophoresis and whatnot. This is a typical result that you'd get. Try not to focus on everything around. This slide is very busy. Just focus on this thing. There's very okay, don't know if I can get that through to you, but these are all super tiny, tinier than the dot I just made, and every spot represents a protein in a sense. And what the robotic station is trying to do is separate all the proteins in the sample into those teeny tiny spots without losing some of the proteins. So maybe imagine separating sugar and salt from each other. You're bound to miss a few bits here and there, but this is tech. It doesn't go wrong. So it's going to separate everything and give you the right number of protein spots in a sense. Just like in the image here. You got a lot of teeny tiny spots all around. And then in the analysis stage, it's going to take every protein and find out how it can affect the other. It gives you this huge network where every small dot represents a protein and its domains, and then it links those to others and whatnot. We're not going to talk a lot about this, but you get the point. It's all about the network. So now you, the smart clinician, what you're going to do is find out if a drug will decrease the amount of a certain protein X here. Let's say over here. So if the drug can decrease the amount of this protein, and this protein is related to another protein's levels, that means that we're killing two birds with one stone. If we get that protein over with the drug, you're also getting the other protein done with. And you're just trying to collapse this network in a sense. And when we say the word collapse network, if you're able to actually do that, the cell is going to die. in cases of cancer and all that, that's of course a good thing. So then you as a clinician, you can also think of things like, okay, we don't like protein Y, it's bad for the patient. Might as well just kill protein X, which is also related to protein Y, using a drug. It's all about this network of relations. This is a huge map that tells you that the small protein here is related to this one and that one and all of those. And that is literally it. Okay, on to clinical value. You can use this stuff, again, just like we said, to know how to control certain proteins in cancer patients. If a protein is bad for the patient, you can try and figure out a way to lower its amounts by finding out if maybe there's a drug that would inhibit the levels of the bad guys, or maybe a protein that the bad guy is related to. Now, you don't just use this stuff to, to know what sort of drug you should administer, but you can also use it to monitor and see if the levels of the protein comes back to normal with the, later on after the drug is used with the patient and all that. Okay, so as for results interpretation, I think that only experts can read those maps, so for now just enjoy the cool colors. They're quite the aesthetic. Even machines know how to match colors. And yeah, that's it. We're done with proteins. We only have metabolites left. I'll take a short break on my side, probably get some, probably get a few other tasks done, and I'll see you in the